why it's doing that. So it's starting to become an issue. So there's a problem with the clarity of some of these images. Oh, good morning, by the way. I don't understand why it looks good on the slide, and then when I put it into Teams, it looks terrible. Richard, have you had any experience with image quality that degenerates in Teams? This one looks okay. And this is fine. And this is great. This one is really fuzzy. Yeah, I don't know either.
We can zoom in and then that makes it clear.
So it's just going to look rubbish. It's going to make my job a little bit more difficult, but hopefully on your screens, is it clear? Yeah. Right. All right. It is what it is. So the first assessment is going to be on Monday. It's going to be a quiz. The quiz is going to be 30 points. Okay. It's going to cover much of the content that's in that chapter, right? But you, having taken genetics with me, are familiar with the types of questions and the format that I put for questions, right? I like to do the following for Cambridge: multiple choice questions, fill in the blanks, true and false, and also short answer questions. Okay, there's a lot of content in chapter 3. Okay, you've covered it all before, and I'm going to test you on some of the key concepts in there, right? And one of those being structure of amino acids. I don't expect you to be able to draw all the configurations, but I do expect you to know which amino acids belong into which groups. Okay? Does everybody have those memorized? You do? Kind of. I put some acronyms on the board that help with memorizing which amino acids are polar, which amino acids are non-polar, and which amino acids are charged. Right? The charged ones are fairly easy to remember. Right? Lysine, arginine, glutamic acid, aspartic acid, and histidine. Right? But the other ones can be a little bit tricky. Okay? What do these letters represent? Single amino acid codes, right? And the order of the letters is a moniker. Okay, so grandma always visits London in May for Winston's party. Okay, if you can remember that sentence and you can remember what the individual letters mean and refer to the amino acids, then it should help you. You just have to run the phrase through in your head a couple of times and you should remember it, right? Grandma always visits London in May for Winston's party. G is what amino acid? Glycine, alanine, valine, lysine, isoleucine, methionine, S, phenylalanine, W, P, proline, right? So those are your non-polar amino acids. Okay? The polar amino acids, this one is serine, but I was going to tell you what the moniker is, okay? Santa's team crafts new quilts yearly. You could come up with any phrase that you wanted that uses those letters as the first letter in the word, right? I just put those up to try and kind of steer you in the right direction, right? And then the last one are the charged amino acids. So dragons eat knights riding horses. Okay, dragons eat knights riding horses. These ones are a bit tricky because the first letter code for the amino acid is not intuitive, right? So, you know, got things like aspartate and glutamate, lysine, arginine, and histidine. Okay, so where we left off, I was starting to talk about protein folding and how about in bacteria, approximately 30% of the proteins require help in order to achieve their final conformation. And in eukaryotes, the number is probably higher than that. It's a guess, but it's around about 50% of proteins, right? And that the help comes in the form of a class of proteins that are called chaperones, right? There are two classes of chaperones, molecular and chaperonins. Okay, they're essential. We find them in all forms of life. Okay. The first set we're going to discuss are two chaperones that are called HSP70 and HSP90. Okay, the HSP acronym comes from their name, which is called heat shock proteins, and they were given that name because of the because of experiments that showed that these proteins, the protein abundance, dramatically increases in bacteria after you heat shock them, and the expression of these proteins is required to help stabilize the conformation of proteins. Okay, so chaperones are not only involved in folding. Okay, some chaperones are involved in activating proteins, and the chaperone will hold on to the protein until it's given a signal to release the protein, and then the protein can go and do its function. All right, so it can act as a switch as well within the cell that turns things off and turns things on. They're extremely abundant. In eukaryotic cells, they constitute a large portion of the proteins that are present within the cell, right? I think the number of copies in eukaryotes is up at around at least 1 or 2 million, which is close to the abundance of ribosomes, right? So they are essential. If you delete these genes, then they are lethal, right? The first class, molecular chaperones, HSP70 and HSP90, work coordinately to help proteins fold, to help stabilize proteins, and to reduce aggregation of proteins, like protein aggregates within the cell that form because of hydrophobic interactions can be dissociated into individual proteins through the action of the chaperones. Okay, this is a highly ATP dependent mechanism. You need lots and lots of energy to be able to make these chaperones change their shape. Okay. HSP70 here. Okay. See the ribosome at the top synthesizing a polypeptide chain. Okay. Chaperones will often associate with the polypeptide as it's being synthesized. And we'll see that happening when ribosomes associate with the endoplasmic reticulum, right? As the protein is being synthesized, then the chaperones attach to the polypeptide chain, okay? Where that's not true for chaperonins, which we will talk about next. Chaperonins work on peptides that have been finished, right? They are complete. The heat shock protein binds to polypeptide, and often we refer to that polypeptide as being the client. The HSP is going to help the client fold correctly, and the polypeptide binds to a substrate pocket on HSP70 in the open conformation. Okay. Then ATP is hydrolyzed to ADP, causing a major conformational change within the heat shock protein. Okay. Now, as well as molecular chaperones, which is the one we're discussing, e.g. HSP70, HSP90, there are co-chaperones as well. And there are, in eukaryotes, at least about 10 to 20 co-chaperones that associate with heat shock proteins. And the co-chaperones help with the hydrolysis of ATP and the change in conformation of the chaperone itself. Okay. The conformational change turns the heat shock protein into the closed conformation, right? So open and then closed. And when it's closed, it holds onto the polypeptide very tightly. Okay. And notice here that the change is caused by hydrolysis of ATP with the help of the co-chaperones. And in bacteria, there are things like DNA K, I believe. What's occurring here is that the binding pocket is holding on to hydrophobic strands within the protein. So stretches of hydrophobicity are stabilized by binding to the HSP, stopping inappropriate hydrophobic interactions from occurring, allowing for the peptide to fold correctly. Okay. Now, in some instances, maybe the protein doesn't fold correctly after this cycle in which ATP hydrolysis is linked to folding. The protein may well come off in an unfolded state, in which case it can enter into another cycle of folding by binding to the HSP or by binding to another heat shock protein, right? HSP90 works in a similar way. However, it's composed of two subunits, not one, like HSP70 is. Okay? Those two subunits form sort of like a clamshell in which binding of ATP maintains the open conformation. Okay? Then when the protein associates, the unfolded protein binds to the holding pocket here, ATP is hydrolyzed to ADP, changing it from the open conformation to the closed conformation when it is stabilized. And at that point, that stable association can just remain like that. And that happens a lot with some transcription factors, where release of the protein leads to proper folding and therefore activation of the transcription factor to go and do its job again, like a molecular switch. The second class of chaperones are chaperonins. Okay, they are much, much larger than chaperones. They are made up of many, many subunits. And in bacteria, the major chaperonin is called GRO-S. G-R-O, capital E, capital S. And the GRO-S is composed of rings of protein subunits. And they also contain caps, which cap the end. This creates a chamber into which the unfolded peptide binds inside. Okay? The lid then caps the end of the chamber, and what follows is a series of ATP-ADP hydrolysis events. It's a closed chamber. You can imagine it a little bit like a salt mill or like a pepper mill, right? Where the bottom cylinders and the top cylinders rotate. And that rotation requires energy in order to make it happen, right? So it's not surprising that these subunits here bind ATP and that conformational changes are fueled by the hydrolysis of ATP to ADP. In prokaryotes, these ring structures are made up of 7 units, 7 subunits, and they're repeating subunits. They're homomeric. So 7 of exactly the same peptide form this ring structure, and another 7 form the bottom ring structure. In eukaryotes, these are a bit bigger, and there's about 9 subunits that are involved instead of 7, and the name changes. So it's a bigger protein. The eukaryote equivalent of this prokaryotic chaperonin is called TRIC, I think. T-R-I-C. So again, we wait until the polypeptide has finished synthesis by the ribosome. And entry into the chamber can occur only in one direction. The other entry point is capped by this protein called GRO-S, right? Sorry, I think GRO-L is the chamber component and GRO-S is the lid. Okay? Then ATP binding to the subunits, we count as 1, 2, 3, 4, 5, 6, 7. And each of these 7 subunits binds a molecule of ATP. And the hydrolysis is coordinated so that they hydrolyze and then turn on the inside. Okay? And we think that the hydrophobic interactions are stabilized, allowing for collapse and then followed by ionic and hydrogen bonding to allow native protein to be folded. Again, this may result in an unfolded state, in which case the protein can enter back into the chaperone to help it refold. The consequences of incorrectly folded proteins can be dire for the health of the cell. Okay? Incorrectly folded proteins mean that they can aggregate within the cell. Okay? And these aggregates are stabilized by primarily hydrophobic interactions between beta sheets. Right? So when they do aggregate, they form a structure that's called an amyloid fiber. Okay? bit difficult to conceptualize, but imagine like a whole bunch of different unfolded proteins all being stabilized by the fact that the hydrophobic residues are shielded. And it creates these long fiber structures that can condense and form into plaques. We see a high abundance of these plaques in patients that suffer from neurodegenerative diseases, things like Parkinson's disease and Alzheimer's disease. The plaques are not soluble, right? Okay. Now we'll move on to protein structure and function. And the example that I'm going to use to describe protein substrate interactions is a well-known model, and that is antibodies, right? We're going to see this repeated over and over again, in that the substrate is recognized specifically by the protein and binds to a region on the protein, right, that's specifically suited for that interaction. The region within the protein to which the substrate binds is called the active site. But you'll have learned in chemistry, in biochemistry, that the active site is just a general term that we use for the site on the protein which binds to the substrate. But in actuality, the binding pocket contains two elements. The first element is the substrate binding site that specifically recognizes the target. The second component of the active site is the catalytic site, where the chemical reaction that the enzyme is going to accomplish is located, right? And so in the model here, right, the substrate binding site here is distinct from the catalytic site. It's contiguous, it's joined to it, right? But they serve two different functions. We'll learn in a little bit using a model for how proteins specifically bind to their substrates. And I'm going to use a class of enzymes that are called proteases. And proteases are enzymes that degrade proteins, right? I'm going to use the example of the pancreatic proteases that are called trypsin, chymotrypsin, and elastase. And they're enzymes that are secreted by the pancreas, which is a secretory organ, into the duodenum, and that helps with the digestion of proteins. Okay? Oh, we all love enzyme kinetics, don't we? Nightmares about that. All right, we'll talk about enzyme kinetics in a little moment, but only briefly and only to support what I'm going to say. I'm not going to ask you to derive a Lineweaver-Burk plot and then calculate Km and Vmax. We won't be doing that, all right? But some of those ideas are conceptually important as we move on. All right, so I'm just going to highlight those. So we've known for a very long time that enzymes speed up reactions, right? And they do so in a very specific manner. Let's see. So at about the turn of the century, about 1900, we understood that the enzyme binds the substrate much like a lock and a key. Okay, that model was superseded about 40 years later, in about 1948, by Linus Pauling. And Linus Pauling suggested that the reasons why enzymes speed up reactions is because they stabilize the transition state of the chemical reaction. So in essence, it's not really that they bind the substrate very, very strongly. It's because they stabilize the transition state, allowing the reaction to move forward, right? And therefore lowering the delta G for that particular chemical reaction. The substrate, often denoted as S, binds to the active site, right? Composed both of the substrate binding site and the catalytic site, and that when the substrate then binds, it forms an even stronger an association because of the stabilization of the transition state. Okay. Two researchers, Michaelis and Mentis, in 1913, did a great deal of work looking at enzyme-substrate interactions. All right, and you'll remember the often penned equation for an enzyme-substrate interaction, right, where E is the enzyme and S indicates substrate. Okay. Michaelis-Mentis were interested in how enzymes are able to catalyze reactions, but they used the initial state of the reaction in order to study it. And so even though this is reversible, okay, at the very beginning of the reaction, right, there's very little product that can turn back. So we can think of it only as the interaction between enzyme and substrate here. Okay. The key thing here is that this enzyme substrate complex can easily dissociate back into enzyme and substrate, right? And so the action of the enzyme is both driven by substrate recognition and the affinity for the substrate. And this affinity is a very helpful variable that describes the enzyme substrate interaction, right? So Michaelis and Mentis showed that in an enzymatic reaction, the initial phase of the reaction is highly dependent on the substrate concentration, right? And that as the reaction proceeds, or in this graph, sorry, we're representing the substrate in increasing concentrations, going from left to right, right? So the linear portion of this graph, which can be defined by the Lineweaver-Burke plot, right, shows that the substrate is limiting until you get to a point in which it doesn't matter if you add more substrate to the reaction, it does not speed up the reaction anymore. And we know the explanation for that. So what is the explanation? Why doesn't this keep on going up like that? Because there are a limited number of sites on the enzyme, right? You can imagine it like, well, the enzyme having one active site to bind the substrate. It doesn't matter if you've got a million substrate molecules. There's only a limited number of sites for the substrate to bind to, right? If you increase the enzyme concentration, right, you can increase the rate of the reaction. A useful measure that comes out of the Michaelis-Menton's graph is that when the reaction reaches its maximum velocity, which is called Vmax, yes, that we can, from the plot, interpret an important commodity, which is called Km. And a Km is the value of the substrate concentration at which we achieve half maximal velocity. Okay? And there's a relationship between the binding affinity and the velocity of the reaction. Yes? So the binding affinity is given by a constant which we call Kd. Right? Another constant for the forward and the reverse reaction is Keq, right? Where Keq is equal to products over reactants, right? Which is in this initial part here, not considering the enzyme product here, but only this part will be products over reactant concentration. Okay. So the important relationship that I want to draw your attention to is that Keq is equal to the inverse of the affinity of the enzyme for its substrate. Okay. So we can see how that would work, right? When the Kd for the reaction is low, it means, so imagine these as being ratios and figures. So imagine this numeral here is small, really small, yes? When you divide 1 by something really small, it's going to make something that's big. So the Kq value is going to be high, yes? If the Kq value is high, it means that products are favored over reactants, right? Representative of an enzyme that has a high affinity for its substrate and would push the reaction forward. Oh, free energy profiles. The only thing that I want to point out in this is that, you know, you have been familiar with reaction coordinate diagrams from like day one. We introduced them in 1107 to describe endogenic and exogenic reactions, right? Things that are, you know, catabolic and anabolic, essentially. Yeah, exogenic would be catabolic, endogenic would be anabolic. But some reactions often have multiple phase changes in them. And so the free energy changes look different from just a single enzyme with a single transition stage here. And I'm not going to go into any bioenergetics. We're not covering mitochondrial metabolism at all, which is usually where it becomes extremely useful to talk about energetics. And we're not going to touch it. So that's all I'm going to say about it. So the model in which we're going to show substrate enzyme interactions is looking at a protease. It's called trypsin-like serine protease. All right, and so these are enzymes which catalytically cleave polypeptides into smaller pieces. And the example that I gave you of digestion of a meal consumed, right? These proteases help to digest proteins that are within the gut, but they're also involved in Other processes such as, which I loved in the book, it's called, moths to escape from their cocoon, they use an enzyme, there's a protease that's called cocoonase. And that's all I wanted to mention because I like that name, cocoonase. The active site of the protease contains both the substrate binding and the catalytic site. Right? Here we have the peptide, which is going to be cleaved in black. Okay. The enzyme here, which is the catalytic site and the substrate binding site, forms a pocket into which the peptide fits. Right? This arrangement here is a parallel beta sheet, right? So it's stabilized by hydrogen bonding between the substrate and the binding pocket, literally holding the peptide in exactly the right position in order to cleave this peptide bond within the backbone of the protein. Right? Now, the specificity of the enzyme comes from this binding site here, which recognizes certain amino acids, right? And so the amino acid that trypsin cleaves next to is an arginine amino acid. These sites are not very specific, right? With the intention that we just want to chop the peptide into really small pieces, right? It's not, in a sense, a one site within the protein that you're going to cleave. This enzyme is going to cleave at every position in which there's an arginine here, and the substrate allows for the binding in the binding pocket, right? It'll literally just snip it up into pieces. Okay. Now, the binding pocket of the enzyme contains this aspartate amino acid, right? And the aspartate is charged at physiological pH, and so is negatively charged. And so it's selective for only allowing amino acids into here that have a long aliphatic chain and a charged group at the end. All right. Other amino acids will not fit into that pocket because they won't be stabilized by binding. Okay. For the other proteases like chymotrypsin and elastase, if you look at the protein sequence and compare the protein sequence of these enzymes, trypsin, chymotrypsin, and elastase, they are remarkably similar in terms of their primary sequence of amino acids. There's very little difference between them. They only differ with respect to, well, the major difference in the catalytic site is this binding pocket here, right? So in chymotrypsin, only really non-polar amino acids are able to fit into this pocket, right? So it cleaves primarily at amino acids that are big and non-polar, like tryptophan, right? Tyrosine, phenylalanine. Elastase, on the other hand, within the binding pocket, normally, so in trypsin, for instance, most of these amino acids that line the pockets are glycines because they've got only a hydrogen, small chain, right? Allowing for arginine to fit into the pocket. Whereas within chymotrypsin, within elastase, those glycines have been replaced by valines. And these valines prevent larger non-polar amino acids from entering into the pocket. So the main target for elastase are amino acids like glycine, right? And so it'll cleave at the next So the next carbonyl join, which is carboxylic acid, joined to the amine in for the next amino acid. Okay. I don't expect you to know in detail the mechanism of cleavage. All right. I'm just going to skim through it, but I don't expect you to know exactly the number of the amino acids and the chemical changes that are taking place in the bonding energies here, right? There are 3 essential amino acids that are involved in the catalytic step, okay? And they're given, you didn't see on the previous one, the catalytic amino acids, they have numbers next to them, right? ASP102, His57, and SER195, right? That means that those amino acids are not next to each other in the polypeptide chain, right? The number refers to the distance from the N terminus to the C terminus, right? So this is aspartate number 102. This is histidine number 57. So the idea here is that these amino acids in the primary chain are really far apart, but when the protein folds, it brings them into close proximity to each other. The main attacker within this reaction is the serine 195. And so the serine 195 attacks the carbonyl carbon, right? These amino acids are referred to as the catalytic triad, okay? Because they're essential. If you change any of these amino acids, then you nullify the protein, right? The transition state is represented by a highly energetic and unfavorable interaction, right? That's stabilized by the protein that's surrounding the point of cleavage, okay? And so this carbon here forms a tetrahedral in which the carbon, is attached to the remainder of the polypeptide chain indicated by X, right, which goes this way, an oxyanion hole, which stabilizes the oxygen, and then also the carbon is covalently bound to the serine, and still the peptide bond remains intact. So that carbon has 4 covalent bonds, right? Next, what happens is that the amine here becomes protonated, and so the point of cleavage afterwards, which is indicated by the Y here, the rest of the peptide floats away, okay? Where this part of the peptide is still attached to the enzyme's pocket through a covalent acyl interaction with the serine. Next, then we want to reset the catalytic site for the next round of proteolysis, all right? And so what happens is a water molecule comes in here, results in hydrolysis of the carbonyl, right? And then release of the second part of the peptide. So what I mean is essentially, if you've got a polypeptide chain like this, right? This was the X part, that was the Y part, and we've cleaved the polypeptide like so. So the X part came off in the first reaction, the Y part came off in the first reaction, and then the X part then is resolved through hydrolysis. one of the amino acids that's part of this catalytic triad is this histidine, right? And its protonated status is really, really important. It has to be deprotonated. And it's only deprotonated at a very specific pH. Yeah, but I think it's pH 6.8. And so if you go above that pH, or if you go below that pH, I'm sorry, then this reaction doesn't work because now the histidine is already protonated and can't attack the polypeptide bond. Which brings me to just about time. There's only a couple of slides left here, I think. Yeah, just 2 or 3. And so the test on Monday will be up to proteasomal degradation, which is the next major topic we're going to cover. Okay. And Tony, I'll send you the test over the weekend. I still have to tweak and modify it. I change them most seasons when I teach the class. Any questions? No? A lot to take in or not?
Particular genetics, your quizzes are making multiple choice. Should we expect fill in the blank and like our test work?
Yes. Yes. And short answers. Not long, just a couple of sentences maybe. I don't want you to, it's only a quiz. You won't be like half a paragraph or anything like that. You'll be okay as long as you do study the material, right? The homework for this chapter I'll release today. I'll try and put as many questions into it as I can. It's not achieved for this textbook is not as fleshed out as achieved for the genetics textbook that I use. And so there's not as much help in the textbook as there is in the genetics book. I'm sorry about that. This is the way it is. Have a great weekend, and I'll see you on Monday. It's okay. I put up when you emailed me, I put up the other one. And so it's easy for me then to put button in just get button clicking or automatically. Then I will do them right after that.
Well, I don't think on the table is flat on the table, like call it.
That's
That's all it was.
Yeah.
All right, first.